So today we're going to talk about the basics of replication, transcription, and translation. Essentially, how do we start with DNA and have that information encoded in the DNA become the information to create a protein? And so today we're just talking about the basics. There's a lot of terminology and jargon in biology, and we really have to have a pretty solid understanding of that terminology in order to understand things like transcription and translation. So this is the flow of information within a cell, and sometimes we call this the central dogma of molecular biology. The word dogma just means accepted truths. So this is the central fact of molecular biology. The DNA can be used to make more DNA in the process of replication. DNA can also be used within a cell as a template to make messenger RNA. Okay, that process is called transcription. And then messenger RNA can be read by ribosomes to create a protein out of amino acids. So this is the central dogma of molecular biology. And this, in essence, is, if you think about it, we have DNA in our cells, and that DNA encodes the instructions for making proteins. And this is the process by which that happens. So where does this all take place within a cell? Okay, well, if we think about it, in our cells, we're eukaryotes. So our cells have nuclei, right? Each cell has a nucleus. Inside of that nucleus is where our DNA is housed. So DNA replication, right? When DNA is going to be replicated to make more DNA, well, DNA is found in the nucleus, so replication is going to take place inside the nucleus. DNA never leaves the nucleus, and that also means that transcription is going to take place within the nucleus. Okay, so using DNA as a template to make a strand of messenger RNA, that's also going to take place in the nucleus. Now, that messenger RNA becomes processed. So that initial copy of messenger RNA from DNA, that actually is going to be processed in 3 stages, and we'll talk about that in a different video. But what's going to happen is, after that precursor messenger RNA, that's the original messenger RNA, after the precursor messenger RNA becomes processed, then we have the final mature piece of messenger RNA. And now that has to leave the nucleus. Okay, so replication and transcription, as well as the processing of that RNA, takes place in the nucleus. But the ribosomes are located in the cytosol of our cells. Okay, they're in the cytoplasm. So that messenger RNA needs to leave the nucleus so that the ribosomes can bind to it and read it. So replication and transcription take place within the nucleus. The messenger RNA leaves the nucleus, and translation happens inside the cytoplasm. So that's where the ribosome is going to bind to that piece of mature messenger RNA, read the sequence found in that messenger RNA, and use that sequence to create a protein out of amino acids, because we know it's those ribosomes that create proteins. Now, this is in a eukaryotic cell. In a bacterial cell, well, bacteria are prokaryotes, so they don't have nuclei. Okay, so each bacterium does not have a nucleus. It still has DNA, of course, like all living cells have DNA, but no nucleus. So we don't have to worry in prokaryotic replication or transcription or translation. That's all going to happen within the cell, but keep in mind that there's no nucleus there to subdivide the replication and transcription parts. Okay? So this is an animation I created just to give us an idea of the basic process of replication, transcription, and translation. So here we have a piece of DNA, a double-stranded piece of DNA, because DNA is double-stranded. And if replication is going to occur, that means that this piece of DNA is going to be replicated. It's going to be copied. Okay, so we're going to start with one piece of DNA, and it's going to be copied. It's 2 pieces of DNA. That's replication. And you'll notice these 2 pieces of DNA are exactly the same. They're identical to each other. So in the process of replication, what's going to happen is that the first thing is that DNA, the 2 strands, have to separate from each other. So here we have those 2 strands of DNA, which are normally held together through the hydrogen bonds between the different nucleotides, right? A, T, C, and G are the nucleotides we find in DNA: adenine, thymine, guanine, and cytosine. So normally when those are base pair together like this, there are hydrogen bonds holding those nucleotides together, and that's what helps to hold the two strands of DNA together. So in the process of replication, those DNA strands separate, okay? And then each of these is going to be used as a template to lay down the new strands of DNA. So here we'll look at the bottom first. Okay, we see that that new strand was laid down, and you'll notice it's exactly the same as the old strand of DNA that's found up at the top of the screen. And that's because we're using the complementary base pairing rules, that on one strand of DNA, if you have thymine, T, then on the other strand, that's going to base pair with A, adenine. And it's the same thing with cytosine on one strand and guanine on the other strand. So the other thing I want you to notice here is when the DNA was laid down, the new nucleotides were laid down in the 5' to 3' direction. And that's how all DNA or RNA is made. It's made starting on the 5' side, and the nucleotides are laid down towards the 3' side. Okay, so 5' to 3'. Let's watch that again, okay? So here's our strand on the bottom that's opened up from the other strand. Here's the 5' end of the DNA. So this strand goes from 5' to 3'. That's the forward direction. And the new strand is going to be made in the opposite direction, 5' to 3'. Now, if you're interested in, well, what is 5', what is 3', what's this forward direction? I have another video about that walks you through why we call it 5' and 3'. But the important thing to keep in mind is that DNA is anti-parallel. So if the bottom strand is going from 5' to 3' on the bottom, that's the forward direction, 5' to 3', then the nucleotides on the other strand are going to go in the opposite direction, right? From 5' on the right to 3' on the left. So it's kind of like a road. You know how one lane of a road goes in one direction? It's forward. You're going forward in that lane. But in the other lane, you go forward in the opposite direction. And it's sort of like that with DNA because it's anti-parallel. So here we've copied that strand of DNA. And the other strand, the original strand of DNA, is going to undergo the same thing. Now, in this case, we see that because this was the other strand of DNA, this is the 3' end, and on the right is the 5' end. So this strand we see here is going 5' to 3', right to left. And that means when this is used as a template to make the other new strand, that the other strand is going to be made not right to left, because the old one here is going right to left. The new strand is going to be made from left to right. Okay, 5' to 3' is going to be left to right. So we see here the DNA has been replicated. Now, this, of course, is just a really small piece of our DNA, and we have billions of nucleotides in human DNA. So this would happen, though, if one cell has to divide and it has to replicate that DNA before undergoing cell division. Now, in the process of transcription, that DNA, the double-stranded DNA, is going to be used as a template to make messenger RNA. Now, DNA is double-stranded, but RNA is only single-stranded. So only one of the two strands of DNA is going to be used as a template to make the messenger RNA. Now, here's the deal. The messenger RNA encodes a gene, and we have thousands of different genes in human DNA, for instance. It's the template strand of the DNA that's used as a template to make the messenger RNA for that gene. And it's not always, with the billions of base pairs we have of DNA, it's not always one strand that's the template strand. It depends on the gene, right? So in this particular example, we have double-stranded DNA here. For one gene, it might be the bottom strand that acts as the template strand. And for another gene, it might be the top strand of the DNA that acts as the template strand for that gene. Okay, but when we're looking at a particular gene, the strand of DNA that's going to be used as the template to make messenger RNA is called the template strand. And that kind of makes sense, right? Because that's going to be the template that's used to make the messenger RNA. Now, DNA is double-stranded. So what has to happen in order for that messenger RNA to be made is that the two strands have to separate so that now the template strand is available. So in the process of transcription, RNA polymerase is going to use the template strand to make, as a template, to make its own RNA, messenger RNA. Now, the same rules apply that in transcription, the nucleotides are laid down in the 5' to 3' direction. So in our template strand here, our template strand is going 5' to 3' is right to left. So RNA polymerase is going to lay down the new nucleotides of RNA in the left to right direction. Okay, left to right is going to be 5' on the left towards this way, 3' on the right. Let's look at that again, okay? See how it lays down the new nucleotides in the 5' to 3' direction. Now, this is the piece of messenger RNA. And you'll notice that if we look at the sequence A, U, G, C, U, A, U, C, G, it was laid down using the same complementary base pairing rules that DNA replication uses, except we know that in RNA, there's no T. There's no thymine. Instead, where there would be a thymine, there's going to be a nucleotide called uracil that's found in messenger RNA. So here we can see, okay, let's just step back one. Here's the DNA. This is T. So we know that in the messenger RNA, we should have an A, adenine, okay, because that's the base pairing rules that T and A pair together and C and G pair together. So we know it would be an A. And now the next nucleotide here that's going to be used in the template strand is an adenine. So in DNA, that would pair with thymine. But again, there's no thymine in RNA. Instead, it's uracil. So that we would put a uracil, a U there. And here's C, right? So C complementary base pairs with G. So the next one would be a G, and so forth. So we can see that's exactly how that happens. Okay, so with the thymine, you have an adenine that's going to be put in the messenger RNA. When there's an adenine in the template strand, RNA polymerase is going to put a uracil in the messenger RNA, right? Cytosine pairs with guanine, guanine pairs with cytosine, adenine pairs with uracil, and so on until the messenger RNA is complete. Now, I want to point out one little trick just to sort of double check that you're doing it right, and just ignore this if it confuses you. But because the RNA uses the same complementary base pairing rules as DNA replication does, if we want to check ourselves, the messenger RNA sequence here should be exactly the same as the DNA sequence on the other strand, except that in messenger RNA, we're going to have uracil wherever there is a thymine found in the DNA. And we can see that, right? A, T, G, in the other strands of the DNA. And in the messenger RNA, we have A, U, G, C, U, A, C, T, A, T, C, G in the DNA. So that's going to be U, C, G in the messenger RNA. So that's a way to check that you've done it right. And that's why this other strand of DNA is often called the coding strand, because it's going to have the same code. It's going to have the same nucleotides in the same order as the messenger RNA, except that we have uracil in messenger RNA and thymine in the other strand of the DNA, right? So we have the template strand. That's the one that's going to be used as a template for RNA polymerase to make that messenger RNA. And then the other strand is called the coding strand, and it's going to have the same sequence as the messenger RNA. Now, after transcription happens and that piece of messenger RNA is made, the DNA, the two strands, are going to anneal again, right? They're going to come together and remain as DNA, because in different cells, we're going to have different messenger RNA that needs to be made. So the DNA is going to open up in the areas where the messenger RNA needs to be made. And after the messenger RNA is made, then the DNA is going to come back and be double-stranded again. So here is our piece of messenger RNA. Okay, and the process of making messenger RNA is called transcription. And it's RNA polymerase as the enzyme that is going to transcribe messenger RNA from DNA. So we have our piece of messenger RNA. It's going to leave the nucleus because this whole process has been taking place in the nucleus. And in the cytoplasm, that's where translation is going to occur. So the ribosome is going to bind to the messenger RNA, and it's going to bind starting on the 5' end. and it's going to scan along and read the sequences of messenger RNA in 3 nucleotide chunks. Okay, those 3 nucleotide chunks are called codons. So in our example, we start with AUG, that's a codon. Okay, CUA, that's the next 3 nucleotides, that's the next codon. And so the third codon here is UCG. So we have 3 codons here. The ribosome is going to bind the 5' ends of the messenger RNA, and based upon those codons, it's going to insert an amino acid. It's going to start assembling a protein out of amino acids, and the amino acid that ends up being in the protein is going to correspond to that 3-letter codon. So AUG, for instance, those 3 letters, that codon, refers to methionine. That's one of our 20 amino acids. CUA is the codon for a different amino acid, which is leucine. And then UCG is the codon for serine. And it continues on and on and on like that. Now, the ribosome is going to scan along the messenger RNA until it finds what's called the start codon. And that's the 3-letter codon that starts the entire protein. And that, as is shown here, is AUG. AUG is the start codon. So when it scans along 5' to 3', when the ribosome finds that codon, AUG, that's when it starts making the protein. And because AUG encodes the amino acid methionine, that means methionine is going to be the first amino acid in our protein. And the ribosome is going to continue after methionine in 3-letter codons, codon by codon by codon by codon, inserting new amino acids, joining that to the protein that it's making based upon what the identity of that codon is. Now, the ribosome does this automatically. The ribosome is using the laws of biochemistry and physics, essentially, to join these different amino acids together. And we'll talk more in a different video about how the ribosome actually assembles these amino acids and selects the different amino acids based upon the different codons that are found in the messenger RNA. But for us to take a step back and look at what these codons refer to, the different codons encode an amino acid, we can use what's called the genetic code. And this is the genetic code. It was actually figured out by scientists who earned a Nobel Prize for determining what codon encodes which amino acids. So we see all the different permutations here, right? Codons have 3 nucleotides in them. So to figure out which codons encode which amino acids, we use the colored boxes on the left, on the top, and then on the right side of this genetic code. So the first base, let's say we have a codon like AUG. To find out which amino acid that encodes for, well, here's our first base on the left. In AUG, the first base is A. So here's A, okay? So we know that somewhere along this row is where AUG is going to be found. The second base of AUG is U. So that's where we go up here to the second base of the codon, and we find, oh, here's U. So A and U, we go down and we find the box where they intersect each other. And here it is, right here. So there are 4 different codons in this box so far. And so on the third base, AUG, we can find it over here on the right side. Okay, we have U, C, A, and G. There's G. So G is going to be on the bottom row of this box. And here it is, AUG. It's actually in green here because that's our start codon. That's where the ribosome is going to start creating that protein out of amino acids. So AUG encodes MET. And MET is a 3-letter abbreviation. We come down here to our key. We see that MET stands for methionine. That's our amino acid, methionine. So the first amino acid is going to be methionine. Now, let's pick out some random one. How about CAU? Okay. If we want to find out which amino acid is encoded by the codon CAU, the first base of the codon is C, and the second base is A. So where these two intersect, C and A intersects right here in this box, and we're looking at CAU. So if we go to the third base, it's U, C, A, and G. We want U, so we know that U is going to be on the top one right here, CAU. And in this case, the codon CAU encodes his. We come down here to histidine, right? His stands for histidine. So the codon CAU encodes histidine, the amino acid. Okay, so you can use this genetic code to figure out which amino acids are encoded for by each codon that you could find. And you can see that there are 64 different possible codons. Just if we think about that, we have 4 different potential nucleotides, U, C, A, and G, right? And they come together in 3-letter codons. We know that there are then going to be, statistically, we can figure it out, that there are 64 different permutations of that. And so here we have the 64, and it's arranged in this little chart to help us so we don't have to just go looking around for AAC, for instance, like, oh, where is AAC in this list? Well, we know A is the first base, A is the second base. So where those two intersect is in this box, and you can use the third side of our code, AAC. Here, C. C is going to be on the second row of that box, and there it is, AAC. AAC stands for, is going to encode the amino acid ASN. ASN is the abbreviation for asparagine. And there we go. So we can use this genetic code to figure out which amino acids are going to correspond to which of the different codons. Now, when we're looking at the genetic code, we see that some of the codons are redundant. And that means that, for instance, let's just take this box here, right? ACU, ACC, ACA, and ACG, those all encode the amino acid THR. And THR stands for threonine. Okay, so any 4 of those codons actually encodes for threonine. So that's why we call it redundant. There are actually 64 different possibilities, but there are only 20 different amino acids. So some of those possible codons are going to encode the same amino acid. Like here, these 4 encode threonine. Now we know that the start codon is AUG. That's going to encode the amino acid methionine. And then the ribosome moves along codon by codon by codon by codon until it encounters a stop codon. And with a stop codon, instead of an amino acid being added to the protein is done. A release factor binds there, and it causes everything to fall apart so that the ribosome releases the protein at that point. So when the ribosome encounters a stop protein, that's its signal that that is the end of the protein, and the protein is released. There are 3 different stop codons, and in this chart, they're encoded by the codons in red. So UAA, UAG, and UGA, those are the 3 stop codons. So when the ribosome is going along codon by codon and encounters UAA, UAG, or UGA in the messenger RNA, that's when it stops and the protein is completed and the protein is released by the ribosome. That is the end of the protein right there. Okay? So those are the stop codons. So again, to sum up the genetic code, this is essentially a key. So if you like puzzles or you like codes like I do, this is kind of fun. It's kind of like you're using this key to try to figure out, okay, if there's this particular codon in the messenger RNA, well, which amino acid is going to be added into the protein based upon that 3-letter codon? So we have our start codon, AUG. That's where the ribosome is going to start creating the protein. Then it's going to go on 3 by 3 by 3 by 3, by codon by codon by codon by codon, until the ribosome reaches one of these 3 stop codons. So this is a summary of the process of transcription and translation. When we have a molecule of DNA, we have the template strand and we have the coding strand. And it's that template strand that's going to be used as a template to transcribe the messenger RNA. RNA polymerase is going to use that template to create a piece of messenger RNA. And then within that messenger RNA, there are going to be codons. Those codons are 3-letter sequences of messenger RNA, 3 nucleotide sequences, and each of those codons is going to code for an amino acid that's going to be put into the protein at that sequence. So here we have 4 codons, codon 1, 2, 3, and 4. AUG is the start codon, and we know AUG encodes methionine. So that's going to be the first amino acid the ribosome puts in the protein. The second codon is CGA. That's going to encode arginine. And we can figure this out using that key, that genetic code. Codon 3 is UCA. That encodes serine. Codon 4 is UGC. That encodes cysteine. And this is just a small piece. So the ribosome would continue along like this in 3 nucleotide chunks until it encounters one of those 3 stop codons, UAG, UAA, or UGA. And that's the process of transcription and translation, just the basics of how this flows. Now, enzymes are going to facilitate the entire process. And in other videos, we'll talk about how transcription occurs with all the enzymes and different molecules that are involved in that. And the same thing with translation. There are different molecules and enzymes involved in the process of translation as well, the process of making that protein.